This conference will now be recorded.
Okay, let's begin. The topic which I will be starting today that with respect to the first chapter is drug-excipient interaction. So clarity is there, voice clarity is there. Asha, can you hear me?
Yes, ma'am.
Fine, absolutely fine. So before going to that topic exactly, I just want to know what is that you know about preformulation. Asha, can you add up any points for this question? What is preformulation means or preformulation studies means? You can just tell your own words. I don't expect any like textbook sentences or definitions. So anyone can answer. Unmute yourself and can answer to this question. What is that idea you have about pre-formulation studies? Try to make the session interactive. If you keep quiet, no, if at all in the classroom, I would have had option of pointing at you and ask the question. But I cannot ask you. So I'm just telling you, giving you option of answering from your side. Chandana. Suchitra. Saundarya, want to answer?
Ma'am, it's a laboratory study to determine active constituent in the excipients.
Don't try to look at the mute yourself. Don't just correlate the try to answer looking at the text, whatever you're seeing on the slide. See, pre-formulation. What is formulation? You can mute yourself. Chitra, you mute yourself. So what is formulation? Designing the, we have an active molecule API, which is already being proved that it has some pharmacological action, and we need to convert into suitable dosage form. So converting that into a suitable dosage form is nothing but called as formulation. Correct. So let me give a simple example at this point. If I want to formulate that drug, whatever already been, we know that it's having its pharmacological action. I want to convert into formulation into tablet. So what I do go with in the initial step, I'll be selecting the excipient or I'll be selecting the or diluents, whatever, and I'll be preparing the granules out of it, and then I'll go for punching. So before punching or before formulating into tablet dosage form, so whatever studies you carry out at the initial stages, pre-formulation studies, that's what. So before formulating into a suitable dosage form, whatever studies we do, that is called as the pre-formulation studies. Example may be like flow properties of the granules which you would have prepared for preparing the tablets, even that is the pre-formulation. Or maybe you're carrying out the drug excipient interaction study, even that is pre-formulation. Or you're characterizing the API itself with respect to its physicochemical properties, like whether it is amorphous state or a crystalline state. So that's what called as the pre-formulation studies. So drug excipient interaction is one of the very important evaluation which you need to carry out before the formulation. So before it helps us to select the suitable excipient for formulating a required dosage form. So it is a before the clinical studies, the critical phase in the drug development before the phase 1 clinical trials. So this is another simple way of expressing the pre-formulations. It includes lot many evaluation parameters that depends on the type of dosage form we are planning. So I cannot just tell in one line, but it is the tests or studies which you carry out before the formulation of any dosage form is nothing but called as the pre-formulation, and generally it comes before the phase 1 clinical trials. So what are the basic things or the general things they do in the pre-formulation is they characterize the physicochemical properties of the API. API is what? Active pharmaceutical ingredient. So they try to characterize, they know each and every details of the physicochemical properties of the drug. As I said, whether like it might be like what is the color, okay, what is the texture, amorphous or crystalline in nature, chemically what it is. So each and every detail, or its solubility, for example, so and melting point, all these details, most more details related to physical chemical properties will be characterized in the pre-formulation studies. You should have defined, I mean, every details should be recorded before you proceed into the formulation stage. And also we'll be looking into the excipients, what excipients we need to select, and that again depends on the dosage form. As I said, when I say diluent in case of tablet, you cannot expect the same diluent when you go with the solution preparation. So when I go with a suspension or emulsion, maybe there I'll be looking at what is the suspending agent or emulsifying agent. So these are the excipients. So it goes on according to the dosage form, the type of excipients also will vary accordingly. So then comes important step is prototype formulations need to be made. Means prototype means which actually we would have planned for a tablet. Exactly the same, whatever we would have planned, the same prototype formulations we are going to make at the lab scale. So that's what generally we do is we do the formulation in the lab scale and then we scale it up. Okay, pilot scale, then comes the production scale. So there's steps which goes on in the formulation developments. We cannot just directly dump. Even in the kitchen, when we plan for cook for 4 members and when we cook for 25 members, our planning will change accordingly. It cannot be the same. So same here even in the pharmaceutical industry, whatever the formulation they do initially in the lab scale, that will be scaled up to the pilot plant scale, which is little large, more like 2 to 3 times more than whatever you would have done in the lab scale, and then it comes the production scale, that is scale up. So pre-formulation is, this is what, so excipient plays very important role in the pre-formulation studies. So initially what they used to do is, that's what I was telling you. So these are some of the basic uh, procedure they used to follow, or the steps they used to follow historically. When you talk about history of pre-formulations, they used to emphasize more on, or more importance they used to give for the bulk characterization. As I was just telling, the same thing is written on the slide. Solubility analysis, stability analysis. Look at the terms inside the bracket. So when I tell about the bulk characterization, so many things comes into picture. Crystallinity, polymer, whether they are polymorphs or amorphous, whether they're hygroscopic or deliquescent in nature, powder flow properties, particle characterizations, like this. So few of them are just highlighted here. So when it comes to the solubility, it's again a big topic where we need to understand the solubility of the drug, not only as the aqueous solubility in water, but also in the different pH, because drug is exposed to the pH environment inside the body, when especially when you take it by the oral route. So it goes into stomach, it get exposed to the acidic pH. So we should know what is happening to the drug at particular pH. So all that in vitro studies will be carried out in the laboratory. Then comes the stability, which is most again important thing which we need to know about the drug. So that helps us to decide about the packaging, how I need to preserve and keep the drug so that its stability will not be disturbed. So its shelf life can be increased. So I need to decide what temperature I have to keep the drug, okay, and what type of packing I'm supposed to do, all that, okay. So that's about the stability analysis. So later it used to come drug excipient interaction. So they initially, historically, when we look into the pre-formulation steps, the drug excipient interaction was not, of course it used to be done, but it was not emphasized so much. But recently now scenario has changed. They're giving more importance for the drug excipient interactions. Okay, so the main objective of the pre-formulation is what? Is to identify compatible excipients. So now the definition is modified accordingly. So it is to identify the compatible excipients, thereby to prevent unnecessary and very costly changes involving time and overall development cost in subsequent steps. So okay, physical property, chemical property, stability aspect of the drug, all that is fine. But if you're failing to identify the suitable excipient, you're going to end up losing the time. You don't get back the time when once you lose it. Okay, and also you'll be losing the money. So unnecessary trials we will be carrying out without understanding whether they're compatible with each other. So then what happens? It results in the loss of time and as well as the you'll be having the that is cost. I mean, you'll be having huge amount of money will be spent throughout that particular study. To avoid that, this is what the main objective pre-formulation has been modified, to identify the suitable compatible excipient so that we can minimize the loss of time, also minimize the expenses incurred with respect to the pre-formulation studies or later on, whatever the expenses. So according to the FDA, when we talk, this process is designated, what process we are talking, pre-formulation, is designated by the term QBD, quality by design it is called as, in which quality is built into the product but not added. We cannot add the quality, we have to make sure that it is built from in each and every step, so that finally we get the quality product. And all that is possible when we carry out each and every step well planned. Okay, and even the drug excipient is part of that. We are need to select a good, proper, compatible, suitable, compatible excipient so that you'll get a good quality product. So that's what I was telling you. Before you plan for any suitable dosage form, I need to come out with a proper planning. So I need to select the probable components that go into the formulation. I need to identify that, and then I need to carry out the drug excipient interaction study. So formulation usually contains a number of additional ingredients that are not API, that is active pharmaceutical ingredient, and these are called as excipients. So directly we cannot take the drug into the formulation to the particular dosage form. I cannot just convert any chemical. Pure drug is nothing but what? It's a chemical form of the drug. So I cannot just convert that drug into a suitable dosage form. I need to add the relevant excipients depending on what is the dosage form. So these excipients actually selected based on the drug excipient interaction study. So this is about an overview of the pre-formulation studies. So what is pre-formulation? These are the studies carried on before the final formulation is done. And this involves basically what? Characterizing the API itself, where we are going to understand all the physicochemical properties of the drug, its solubility, its stability. Not only that, we are also going to identify the suitable inactive ingredients, that is the so-called as additional ingredients, or you call it as excipients. We're going to identify the suitable excipients also, and that is very much needed because that can minimize your time, okay, time or the wastage of money and all that. The negative aspects can be overcome by proper planning of your studies. So let's look into what are these excipients mean. So though you have been using this term throughout your BPharm UG syllabus, so let's understand what are excipients. So what is the meaning of inert word here? Tejaswini?
Yes, ma'am.
What is the meaning of inert? They're considered inert. Excipients
Non
are,
non-active, ma'am.
yeah,
Non-reactive.
yeah, not, doesn't have any pharmacological activity. For example, when I take paracetamol, it's an antipyretic, right? If I add an excipient, if it also has an antipyretic activity, then things, or it might have a some other activity, if at all, if it has some other activity, then that results in the unwanted effect. So they are the inert. Excipients are nothing, no doubt, they are also chemicals. Okay, even API is also a chemical. But these excipients are inert with respect to the pharmacodynamic activity. They are inert. It doesn't have, usually doesn't have any kind of pharmacological actions, but they do have some important functions. Okay, they are important in the manufacture of the drug product, and it can provide functionality to the drug product with respect to drug release and dissolution. Though they are inert in nature, but they do play a very important role. This you have to remember. They add up functionality. I'll give you the example in later slide. They add up functionality to the drug product, and also it can also alter the release mechanism. The drug should be released immediately, or the drug has to be really released in a sustained manner or a controlled manner. So all this can be controlled with the help of the excipients. So, and also it can affect the drug product performance in vivo. Means, for example, if I give the, at this point, enteric coated tablet, if I give the example, it plays a very important role when we talk about the gastric irritation, because enteric coating prevents the drug causing some causing gastric irritation. So because it's forming the code. The excipient is forming the code. So the in vivo performance of some of the formulation can also be altered by adding the suitable excipient. Means though we are calling in the first statement, they are inert, doesn't have any pharmacodynamic or pharmacological activity, but they do have add up some required functionality to the drug product design. Okay. So that's what I was telling you. These are the list. When you look at the functional properties of the excipients, what all the important functional properties can be exhibited by the excipients? So it can improve the manufacturability of the dosage form. For example, you add magnesium stearate, the lubricant and glidant. If you remember the tablet manufacturing procedure, why we add that glidant and lubricant? So that the you get the desired flow property. So it can flow easily from the hopper and get filled into the cavities, and so there'll be no weight variation or there'll not be any content variation. So it improves that, improve the manufacturability of the dosage form, stabilize the drug against the degradation. Of course, even that can be expected. You can add as a stabilizer, decreases gastric irritation. Just now I gave you an example. Control the rate of drug absorption from the absorption site. So depending on the polymer, whatever we add, which is nothing but the excipient again, I can actually control the drug release from my dosage form however I want. Immediate, for example, if I take an immediate release tablet, it follows a simple sequence. As soon as I take with a glass of water a tablet, after some minutes or within 10 minutes, it undergo disintegration, okay, and then it undergo dissolution, followed by that is the absorption. So within half an hour to 45 minutes, you can expect the absorption to happen. So but those are called as immediate release, IR release dosage forms. But when I say modified release dosage form, modified, so I'm modifying the release accordingly. I don't want the drug to be released immediately. I want the drug to be released throughout the day or throughout the night. So what I do, the added excipient I select such that it will not allow the drug to release immediately. So it will start releasing the drug. So whatever topic you would have learned in the control delivery systems in your Pharm UG 8th semester, that knowledge you can apply here. So later one is the increased drug bioavailability also. So if I can prevent the degradation of the drug happening inside the body, definitely its bioavailability can be increased, because I'm not giving chance for the drug to get degraded in the body, maybe because of the enzymes present in the GI tract or maybe the pH change in the GI tract. So I don't want my drug to be degraded when it reaches that particular GI tract. So I can avoid that and thereby increase the bioavailability. That's one of an example I'm giving. So there are different approaches by which by adding the excipient I can enhance the bioavailability. Maybe I can add an excipient which increases the solubility of the drug. By increasing the solubility of the drug also I can expect the good bioavailability. So whatever the strategies you might apply, but I'm just giving you an idea, the overall the functional properties of the excipients, even though these excipients are inert in nature, they play very important role in the dosage form designing. Not only in vitro during the formulation, that is outside the body, in vivo also they play very important role. So drug excipients in the drug product may also affect the dissolution kinetics of the drug. That's what I was saying, control, sustain or prolong, whatever you call that, depending on the kind of release. So that affects the kinetics. First order kinetics it might follow, or a zero order kinetic it might follow. So that can be decided based on the excipient we are adding. For example, matrix type tablet or core type tablet, where the drug will be inside the core and surrounded by the polymer, and how the drug will come out by the diffusion mechanism. So there's so many things to understand. So this is an overall picture for your knowledge that the added excipient can add up many kind of functionality during the formulation of a particular dosage form. So coming into the actual topic is the drug excipient interaction. I told you, if you can select a suitable excipient in the beginning stage itself, so that will actually avoid all the negative aspects of selecting a wrong excipient. See, maybe I would have not done a proper study of the drug excipient interaction. Nothing goes wrong in the lab scale. Even the pilot scale, I may not see any kind of problem. But maybe when I take it to the that is production scale, that's when it might start showing the interaction. And at that point, you can imagine so much of money you would have invested, so much of time you would have spent in planning for that. All that will go waste. So to avoid that, in the pre-formulation study, they're emphasizing, they're giving so much importance about the drug excipient interaction that carry out the drug excipient interaction, spend some time. It's okay. There's nothing wrong in spending some time, but select the suitable excipient so that you'll not be wasting time and money later part. So elucidation of possible chemical or physical reaction in a formulation is an absolute requirement to establish the stability of given dosage form. So ultimately, it is a dosage form designing you would have successfully done. If it is not stable, the stability of the dosage form may be affected because of many things. Because the drug will be interacting with the excipients, or maybe the drug is interacting with the packaging material. So there is a lot many possibilities, or maybe the drug is not stored in the suitable required temperature condition. Okay, so anything, if it disturbs the stability of the dosage form, by the time it reaches the consumer, that is we are the consumers, okay, or customers. So by the time we use it for our need, if its stability is lost, then everything is waste. So you need to elucidate the possible chemical or the physical reaction in a formulation, which is an absolute requirement to establish the stability of given dosage form. So ultimately, to have a good stable product till it reaches the customer or during its shelf life, we need to always carry out the or elucidate the possible chemical and the physical reactions. So these reactions can be of many types, which I'll be discussing again in detail later on. So all the possible solid-solid reaction of a drug with excipient should be established for the completion of pre-formulation studies. So the time is exclusively dedicated in the pre-formulation studies for identifying any kind of solid-solid reaction. So solid-solid reaction, or it depends again, no, it depends on the type of dosage form you're preparing. So if I take tablet, most of all the ingredients are solid in nature. So you might feel many times that, okay, solid may not interact with another solid, but there is a chance there is a solid-solid reaction, which can happen because of the environmental factor. It can be humidity around it, or it can be the temperature around it. So like this, or it can be the oxygen, the oxidation. You would have learned about the degradation by the oxidation process. So all the possible solid-solid reaction of a drug with excipient should be established. So what they do is they identify the API, okay, fine. Then they start looking into the excipients. So maybe I have 100 excipients in my hand. Okay, based on literature search, I would have got. Okay, and now I'm not going to find out the interaction with all the 100 excipients. It's again the waste of time. Okay, that's when your optimization chapter comes into picture, which you'll be learning in detail in the second subtopic of first chapter, how this optimization technique will help to reduce screening or reduce the selection of the excipient from 100 to 10. So that will minimize your time again. So you'll be saving your time and money because of optimization technique. So I'll be selecting some 10 to 15 out of, or maybe the 20 or 30, whatever. The narrowing down the number from 100 to this many number is also important step. Then I start analyzing. Okay, for example, I will select disintegrant, I'll select diluent, I'll select lubricant. So I'll be selecting 2, 3 based on my literature search, which would have justified in the literature that it's having a magnesium stear is good, or the lactose is a good bulking agent. So based on the literature search, I would have selected now. Now I start looking at the other angle, that is whether my drug is incompatible with that of the selected excipient. So that's what called as solid-solid reaction. This is nothing but a part of drug excipient interaction. So this, when you talk about the interaction, there is a variety of interaction can happen. So interaction can, the drug substance may undergo a variety of interactions or transformation by one or more physical or chemical ways or methods. So, okay, now the drug excipient reactions can happen physically or chemically. So when you talk about the solid dosage form, see, I'm stressing upon the solid dosage form. So because when I tell liquid dosage form, it's not a solid, definitely. So there is an instability issues with respect to liquid component interacting with the soluble component. So that's a different thing. So when you're talking about the solid dosage form, is the interaction happening between the solid and solid, and that interaction can be a chemical change or the physical. So it can be a chemical interaction or a physical interaction. So interaction study must be approached rationally to avoid unnecessary investment and time. So that's what I was telling you. Just because it's been emphasized so much that drug excipient interaction need to be studied, I cannot just waste time there. So I need to be rationally thinking how to do that so that I'll not be wasting my time and money at this point also. That's what I told you. There are different approaches they usually follow, and they narrow down to the required number of excipient, and then they start with the drug excipient interaction. So we need to always give importance for the drug excipient interaction, keeping, I mean, irrational, not irrationally. So however I want, I cannot just do, I need to properly plan for that. So what happens when the drug interaction happens with or the drug excipient interaction happens? These are the possible changes which can happen. So it can result in the formation of the new impurities. Impurities, when we talk about the word impurity, the impurity can be added up in the raw material itself. Even the quality checking will be done at the raw material when the raw material initially comes for the before the production purpose. No, they will scrutinize that. They will actually look into the quality of the raw materials. There is a chance that impurities which are already been present in the bulk material, but if that is taken care, okay, the bulk material is no impurity is present, but there is a chance that now after adding the excipient, the new impurities might add up because of the interaction. So there might be a formation of new impurities. So there can be incomplete mass balance. See, add something, I should get same thing out. I add 4, for example,